If someone insisted that they could feed everyone on this planet and house and clothe them and cure their ills using nothing but air and water, you'd be completely skeptical. And if you're thinking no one would make such extraordinary claims, you're right. Not because these are impossible to achieve, but because plants can't say much of anything. Silent and unmoving, plants can seem just a pleasant backdrop to the comings and goings of animal life. But it's plants that hold the secret to capturing solar energy and using it to build biological molecules, literally out of air and water. An estimated 450 gigatons of carbon, that's 450 followed by 9 zeros in metric tons, are in plant biomass, built by plants beginning with air and water. And the roughly 8 billion of us humans? Well, our biomass is a relatively puny 100 million tons. And we're here at all only because of food made possible by a crucial set of reactions that happen in the chloroplasts of plants. In photosynthesis, plants convert light energy into chemical energy and ATP, producing NADPH, which gains electrons for driving anabolic reduction reactions. That first phase of photosynthesis is also called the light reactions, and it supplies plants with two important molecules, NADPH and ATP, which they need for the second phase where sugar is synthesized. In this lecture, you will learn how plants use energy and reduction reactions to build carbohydrates from carbon dioxide and water. This alchemy, making food from air and water, occurs in a series of reactions called the Calvin cycle. You'll learn about the world's most abundant and arguably most important protein and how its inefficiency limits photosynthetic yields. We'll then take a look at how some plants have mechanisms to deal with this problem. And you'll see how researchers have recently found an ingenious workaround for plants that don't have their own solutions, opening a door to improving crop yields immensely. But just as one does not live by bread alone, plants provide us with far more than essential nutrition. Our lives are also enriched by a multitude of other plant molecules called secondary metabolites. These molecules are not necessary for plant growth and reproduction, but we'll take a quick tour of these molecules, which include, among other things, flavors, dyes, caffeine, and even catnip. So let's start with photosynthesis. As we just saw, the Calvin cycle constitutes the second phase of photosynthesis, where the light energy is used to build up the molecules that make up the plants, support their growth, and feed the rest of the world. Let's take a minute to first think about what's happening in biochemical terms. Carbon dioxide is being reduced, chemically speaking, to a carbohydrate. You know that when a molecule is reduced, it accepts electrons. You also know that a carbohydrate is a more complex molecule than carbon dioxide. So the Calvin cycle involves a gain of electrons by carbon dioxide and an increase in complexity, meaning that a source of electrons and a source of energy are needed. Another name for what's going on is simply carbon fixation. With that as an overview, we can look more closely at the reactions of the cycle. The first of these is the addition of carbon dioxide to a 5-carbon phosphorylated molecule called ribulose 1,5-bisphosphate, or RUBP. This reaction is catalyzed by the enzyme everybody calls Rubisco, which is short for RUBP carboxylase oxygenase. You see why. The Rubisco enzyme is the most abundant protein in the world. In the leaves of plants, it may make up as much as 20 to 50% of the total protein. Whenever you eat a green salad, you eat some Rubisco. And as the catalyst for the first reaction in making food from carbon dioxide, Rubisco is an enzyme that the world depends on. For an enzyme of such central importance, Rubisco, though, is dreadfully inefficient, as we'll see in a minute. So what is the product of the Rubisco reaction? Remember, we added 1 carbon dioxide to a 5-carbon RUBP. We get a 6-carbon compound called 3-keto, 2-carboxy-arabinitol, 1,5-bisphosphate. That molecule turns out to be extremely unstable, which is a blessing to anyone worried about having to remember its name. When it breaks down, it splits into two identical molecules, each containing 3 carbons known as 3-phosphoglycerate or 3-PG. If that name sounds familiar, it should, because all living organisms make this molecule in glycolysis and gluconeogenesis. At this point, the first of 3 stages in the Calvin cycle is done. In the next stage, you'll see that the 3-PG is reduced to make the molecule glyceraldehyde 3-phosphate in reactions that use ATP and NADPH from the light reactions. NADPH donates the electrons needed for reduction, and ATP provides the energy. Notice that the reactions taking 3-PG to G3P are identical in steps in gluconeogenesis. This makes some sense as both pathways synthesize sugar. But in the Calvin cycle, the reactions do not go on to make glucose, not directly anyway. Getting to glucose is a bit more complicated because so far only 1 carbon dioxide has been taken up. Only after running the cycle 6 times are there enough carbons captured to make a 6-carbon sugar like glucose. To continue running the cycle, some of the G3P molecules are recycled to make more RUBP. Regeneration of RUBP is the 3rd stage in the Calvin cycle. And RUBP from the 3rd stage is also the molecule that reacts with carbon dioxide in the very first reaction of the cycle. This 3rd stage has several possible inputs and multiple outputs, but we're going to skip over those wider considerations and focus only on the regeneration of RUBP. How much we get depends on the availability of ATP from the light reactions. Okay, so it takes 6 turns of the Calvin cycle to get enough carbon to make a glucose. Each complete turn requires 3 ATP and 2 NADPHs. So the 6 turns needed for making a single glucose molecule requires 18 ATP and 12 NADPHs. And these, as we know, are made in the light reactions. The G3P made in the Calvin cycle has 2 fates. Some of it is siphoned off from the cycle to go into making sugar. And some of it goes back into making RUBP to continue carbon dioxide capture. And that, in a nutshell, is how photosynthesis makes food out of water and carbon dioxide. Now, don't forget, water enters the picture in the light reactions. It's water that provided the electrons for the electron transport chain in chloroplasts to make NADPH and indirectly ATP. Splitting water also was what released the oxygen. Now, of course, plants don't make food or produce oxygen for our benefit. Plants need glucose for their own cellular reactions. Extra glucose is stored as starch in tubers like potatoes and in the seeds of grain plants like wheat and rice. Plants also use glucose to make cellulose. Cellulose, as you have learned, is a polymer of glucose that makes up the plant cell wall. It's also the primary constituent of cotton fibers. The production of oxygen during photosynthesis is useful to us, but it also has some unintended consequences. Early photosynthetic organisms functioned in an atmosphere that contained much less oxygen than we now have. But oxygen levels rose as photosynthesis kept pumping it out. This made it possible for a lot of new oxygen-dependent organisms to thrive. But that created a problem for photosynthesis. It turns out that rubisco, the enzyme that captures carbon dioxide from the air, is not very good at distinguishing oxygen from carbon dioxide. About a quarter of the time, rubisco accidentally binds oxygen instead of carbon dioxide. Consequently, there's no carbon dioxide added to the 5-carbon RUBP. So instead of getting a 6-carbon intermediate that splits into 2 molecules of 3-PG, we get 1 molecule of 3-PG and a 2-carbon molecule called 2-phosphoglycolate. This unfortunately reduces the efficiency of carbon dioxide capture. Instead of 2 3-PGs, we get 1. And even worse, 2-phosphoglycolate is toxic to cells, so it's got to be dealt with. Plants manage this through a series of reactions called photorespiration that convert the 2-phosphoglycolate into 3-PG. But this recycling costs plants more than 12 ATPs per molecule of 2-phosphoglycolate. And in the process, some of the carbon is lost as carbon dioxide. Moreover, getting oxygen confused with carbon dioxide becomes even more likely when temperatures rise. So this problem is going to get worse as the world warms up. Now, plants are in a fix regarding photorespiration. It greatly reduces the efficiency with which the Calvin cycle runs, but they also need it to detoxify 2-phosphoglycolate. So it's obvious that finding ways around photorespiration would be worthwhile. In fact, not all plants have just been sitting around photosynthesizing. Some plants have come up with interesting fixes. Most plants directly capture carbon dioxide from the air, combining it with RUBP, as we've seen, to produce 3-PG. Because this inefficient approach to carbon capture directly creates a 3-carbon molecule, these plants are called C3 plants. Some flowering plants have evolved a different carbon fixation approach to reduce the oxygen problems. The leaves of these plants have a specialized anatomy that separates the capture of carbon dioxide from the reactions involving rubisco. So one set of cells called the mesophyll, closer to the leaf surface in these plants, is in contact with atmospheric CO2. But unlike C3 plants that use rubisco to take carbon dioxide directly from the atmosphere, these plants use a different enzyme, PEP carboxylase. It combines the carbon dioxide with phosphoenol pyruvate to form oxaloacetate. Because this alternative approach to carbon capture yields the 4-carbon oxaloacetate, these plants are called C4 plants. The C4 pathway is used by plants like prairie grasses and sugarcane, which grow in hot places where photorespiration would quickly dry them out. But how does this help? Well, if you want your kid to eat healthy foods and not fill up on cookies, you don't send her to the buffet to help herself. You sit her down at the table and you bring her a plate with only healthy stuff. That's sort of what C4 plants do. By not using rubisco to capture the CO2 initially, C4 plants avoid rubisco's mess from binding to unwanted oxygens. Instead, C4 plants keep the rubisco tightly packed in another set of cells called the bundle sheath with less access to oxygen. The oxaloacetate in the mesophyll, meanwhile, is converted to malate, another 4-carbon molecule, which gets transported to the bundle sheath and then is broken down to release the carbon dioxide right where the rubisco is. And by bringing the carbon dioxide to the rubisco, rather than letting it help itself from the atmosphere, where the oxygen concentration is high, C4 plants reduce photorespiration greatly. So C4 plants solve the photorespiration problem by keeping enzymes for carbon dioxide capture in different parts of the leaves from where the rubisco is located. Another solution to the photorespiration problem evolved in plants native to very hot and dry places. Their adaptation is called crassulacean acid metabolism, or CAM, named for the Crassulaceae family where this was first discovered. CAM plants include orchids, cacti, and pineapple. They use the C4 strategy of reducing the need for photorespiration, and they also save water. To understand how they do both, leaves on their surfaces have pores through which gases and water vapor diffuse. These pores, called stomata, can be opened and closed by the plant. CAM plants, which are adapted to desert conditions, open stomata only at night when it is cooler and the humidity is higher. This reduces water loss, but it also limits when carbon dioxide is available to the leaf cells. At night, carbon dioxide diffuses into the leaf and is captured by PEP carboxylase to make oxaloacetate, just like the C4 plants. This is then stored in sac-like structures called vacuoles in the cell until the next morning. When the sun comes up, CAM plants close their stomata, limiting the entry of gas, including oxygen. Then they break down the oxaloacetate in the presence of rubisco, just like C4 plants, to release carbon dioxide. Because they release all their carbon dioxide during the day while their stomata are closed and oxygen is not being admitted, these plants too are able to surround rubisco with carbon dioxide and avoid binding to oxygen. So we've seen that these two groups of plants, the C4 and CAM plants, evolved adaptations that compensate for rubisco's inefficiency. But unfortunately, most plants are C3s, and in these plants, photorespiration reduces crop yields significantly, by more than a third for soybeans, for example. A 2016 estimate indicated that reducing photorespiratory loss by just 5% would be worth over $540 million a year in the Midwest. But this is an enormous challenge. It is not easy to engineer C3 plants to behave like C4 or CAM plants, given that C4 plants have a very different kind of leaf anatomy, and CAM plants control their stomata to limit when they gain access to carbon dioxide. These systems are entirely different from those in C3 plants and cannot be readily transferred into them. Now, given these obstacles, researchers wondered whether photorespiration could be made less of a drain on the system. As we saw, photorespiration involves a series of reactions to detoxify 2-phosphoglycolate and recover 3-PG in the process. But besides the fact that it is an energy hog and not good at salvaging carbons, it is inefficient in another way. Like a nightmarish bureaucracy, the detoxification pathway is made up of some 9 enzymatic reactions spread in all different parts of the cell. Here's a quick overview. The 2-phosphoglycolate made by rubisco is converted to glycolate. And then this gets sent to another cellular compartment, the peroxisome, where it gets converted to glyoxylate and then to glycine. And then a series of 3 more reactions and 3 organelle changes results in the deposit of a 3-PG replacement back in the chloroplast. This process is energy intensive, and along the way, a carbon dioxide is lost. The researchers' goal was to shortcut the cellular bureaucracy by engineering plants to detoxify the 2-phosphoglycolate directly in the chloroplast. An immediate advantage would be that no CO2 would be lost and less energy would be used. In 2019, researchers were able to do just that. By introducing the genes for just 2 enzymes into chloroplast, they bypassed photorespiration. Now, what did these enzymes do? First, one was an enzyme normally found in peroxisomes that converts glycolate into glyoxylate. The other is normally found in another plant organelle that adds acetyl-CoA to glyoxylate to make malate. Remember what C4 plants do with malate? Yes, they use it as a source of carbon dioxide, which in this case is released within the chloroplast where the rubisco is. To ensure that this pathway for recycling 2-phosphoglycolate remained in the chloroplast, scientists also inactivated the transporter that normally moved glycolate away from the chloroplast to the peroxisome. With nowhere to go, the glycolate was dealt with inside the chloroplast. And best of all, in field trials of these modified plants, yields went up by more than 40%. Now, this extraordinary jump in productivity contrasts sharply with what's possible through conventional crop breeding. Most breeds are already well optimized, so improved breeding typically generates less than a 2% increase in yield, even after many years. With the population projected to increase by as much as 30% by 2050, the world is going to need large increases in food production, and plants with a little help cleaning up photosynthesis may be able to help feed all of us. Having looked at the primary things that plants do for us, food and oxygen, now let's briefly examine 3 families of secondary metabolites that plants also make. By definition, secondary metabolites are not essential for plant growth and reproduction, but they often serve to protect plants against pests. It just so happens that many of these molecules are also valued by humans. Let's begin with alkaloids and their most notable member, caffeine. Caffeine is biochemically a methylxanthine, and that's a purine compound similar to the bases adenine and guanine in DNA and RNA. Indeed, caffeine is derived from the purine nucleotides. Let's remember that nucleotides contain a base, in this case adenine or guanine, a sugar, and one or more phosphates. Both adenosine monophosphate, AMP, and guanosine monophosphate, GMP, can give rise to a related molecule, xanthosine monophosphate, XMP. Now, XMP loses its phosphate group to become xanthosine, which contains the base xanthine and a ribose sugar. From there, 3 methyl groups, CH3, are added, and the sugar is lost, giving us caffeine. Caffeine binds adenosine receptors in the brain. This isn't terribly surprising, given that caffeine is structurally similar to adenosine. But what does that do? It turns out that adenosine normally accumulates during our waking hours and binds to those brain receptors. Adenosine receptors filling up with adenosine is a signal to our body to slow down and rest. This is why you get sleepy after a long day. When you sleep, adenosine levels go down, emptying the receptors and eventually leading to wakefulness, except maybe on Monday morning. Caffeine binds to those adenosine receptors, but it doesn't make you sleepy. And because adenosine can't bind when caffeine is occupying its receptors, you stay alert. If you drink coffee regularly, however, the brain compensates by making more adenosine receptors. And when that happens, you need to drink more coffee to fill up the adenosine receptors and stay awake compared to someone who didn't start drinking coffee. You may have noticed this. Another group of secondary metabolites is the phenolic compounds, all defined by a phenol ring structure. Examples include the main flavor of vanilla and salicylic acid, the starting molecule for aspirin. Phenol is a benzene ring with a hydroxyl group attached. Plants make a bewildering variety of phenolic compounds, including polyphenols, which can have multiple phenol rings. But they all start from the amino acid phenylalanine. Vanillin, the main flavor component of vanilla, is synthesized by the vanilla orchid, but natural vanilla extracts contain small amounts of hundreds of other aromatic compounds that give it complexity. Artificial vanilla is pretty much pure vanillin and is made from, I kid you not, byproducts of the wood pulp industry. Now this may seem weird, but it's not as weird as it might seem because lignin, a major component of wood, is made by the same biosynthetic pathway as vanillin, starting with phenylalanine and tyrosine. Cinnamon gets its sweet and spicy flavor from cinnamaldehyde, a molecular relative of the phenolics. So far, cinnamon is still obtained from the bark of the cinnamon tree. A fascinating study published in 2017 shows that cinnamaldehyde may help fight obesity by increasing production of uncoupling protein in the fat cells of mice. Now you might remember when this happens, as you saw in the last lecture, the burning of fat does not generate ATP, but instead is dissipated as heat. There may be side effects that we don't know about, so don't dump a ton of cinnamon in your food until scientists figure out what's safe. Some phenolic compounds cure your aches and pains. Salicylic acid, made by many plants but found in large quantities in the willow, is also derived from phenylalanine. Salicylic acid works by inhibiting the COX enzymes that synthesize prostaglandins, the signaling molecules that mediate inflammation. Salicylic acid in willow extracts was used in folk medicine for many years, was the forerunner of aspirin, which is the derivative of acetyl salicylic acid. Another phenolic compound of considerable interest is resveratrol, a metabolite found in grapes, red wine, and some berries. What has many people excited is that feeding resveratrol to experimental animals, from worms to fruit flies to fish to mice, increases their lifespan. Finally, I'd like to touch on a group that gives many herbs and spices their characteristic aromas, the terpenes and terpenoids. The fresh, piney smell of evergreens comes from pinene. A scent of lavender comes from linalool. And citrus fruits get their overall smell from limonene. And the precious resins, frankincense and myrrh of the Christmas story contain a mix of terpenoids that contribute to their fragrance. Terpenes are hydrocarbon compounds often manufactured using resin from evergreen trees. This is why the name terpene sounds like turpentine. Both are derived from pine tree resin. There are a couple of different biochemical pathways to terpenes, including one beginning with mevalonate, the same molecule that animals use to make cholesterol. Terpenoids are modified terpenes that add other groups, often containing oxygen. Now, also included in the terpene group are the carotenoids, of which over 1,000 are known. Carotenoids are pigments that make carrots orange and corn and daffodils yellow. In plants, carotenoids serve as accessory pigments. They absorb different wavelengths of light than chlorophylls, but are able to pass on the light's energy to chlorophyll. So they extend the range of wavelengths that can be used for photosynthesis. Now for us, as for Bugs Bunny, the carotenoid beta-carotene is the starting material for making vitamin A. Our intestinal cells convert beta-carotene into retinol, one form of vitamin A. Two other carotenoids, lutein and zeaxanthin, found abundantly in green leafy plants like kale and spinach, are the only pigments that get deposited in the macula, the central portion of our retinas. There they apparently safeguard retinal cells against oxidative damage and protect against age-related macular degeneration, the principal cause of blindness in people over 65. Aromas and pigments. What's next? Well, plants make many more terpenoids than we could possibly discuss, including those that make up latex rubber and amber. But we'll end our exploration of plant metabolites with two molecules. One that satisfies our craving for sweet tastes, and another that makes our feline pets happy. The terpenoid that's a sweetener molecule is called ribadioside A. This odd molecule is responsible for the sweet taste of the sugar substitute stevia, made by the leaves of the plant Stevia rebaudiana. The structure is a diterpene glycoside, which is to say a diterpene, that's the 3 hexagonal rings in the middle, plus a bunch of glucoses attached to it. The result is over 250 times as sweet as table sugar, sucrose. It's called a non-nutritive sweetener because the glucose sugar units in it cannot be metabolized by humans for energy. Unlike other artificial sweeteners, ribadioside A has been shown to stimulate insulin production by the pancreas without requiring increased blood sugar to do so. More research is needed to understand how this terpenoid works, but it is potentially good news for people looking to control blood sugar. And finally, there is the terpenoid known as catnip. Flowering plants of the Nepeta genus make this molecule called nepetolactone, which apparently has an effect similar to sex pheromones of cats. Big cats seem to be turned on by it too. So why would plants make molecules to seduce cats? They don't. Nepetolactone is a powerful insect repellent, so you could use it to keep the insects at bay. Just be sure there's no tigers in the area. Let's wrap up with a plant wrap. Tiny plants with tiny stems, our source of life. Let's say amen. They take up water, air, and then release back tons of oxygen. C3 plants with rubisco bear grab CO2 without a care. But there's a problem, beware. O2 is oft what's really there. C4 plants assimilate with phosphoenol pyruvate, then transfer with a small malate, avoiding making glycolate. The CAM plants close stomata tight and take up CO2 at night, then hold it till the morning light to do the Calvin cycle right. Which brings us to the final scene, the quest to make the planet clean. Perhaps we'll use a plant-based scheme, because they have solar panels green.